Great, thank you. So greetings from Cork in Ireland, and this is a picture of Cork Harbour, just down the road from me here. I'm delighted that everyone can join today. I know that there are people from all around the world, so it's lovely to be reaching out to you today from Cork in Ireland. And a huge thanks to GloFouling and to Jurga and to John for inviting me to do this presentation. So very briefly today, in the next 20 minutes, I want to spend a little bit of time talking to you about a few key elements, mainly that marine diseases are increasing. There are a number of reasons for this. I'm going to touch on that briefly. That movement and establishment of invasive organisms are increasing. And this is actually becoming one mechanism that is facilitating the spread of parasites, pathogens, and disease in marine environments. It's often quite challenging to make this link conclusively. And I suppose today I want to spend a little bit of time in demonstrating some aspects of our work on shellfish, on aquaculture diseases, and on invasive aliens and how they might all be linked up. So I suppose the first thing I just want to focus on is that the incidence of marine diseases is increasing. And this is due to a number of factors. There's increased activity in the marine environment, and this is linked to the introduction of new hosts moving to new habitats and new areas. And in association with this, and often independently as well, this can result in the introduction of new parasites and diseases. There are other factors that are also responsible for this increasing incidence of marine disease. I'm not going to really talk about those today, but things like habitat loss or stress and climate is becoming a bigger factor all the time in these increasing incidences. When we look at some of the mechanisms that invasive species use to move around, particularly not just in marine systems, also terrestrial, but when we look at marine systems, the main pathways are via transportation. Many of you will know about the impact of fouling on ballast water release, on living industries such as aquaculture, and there's a whole range of miscellaneous pathways and intentional and unintentional movements of organisms around the world. Now, there's quite a lot of material on this slide, but if you look at the map of the world on the right-hand side, and you can see different colors around some of the various countries, particularly when you look around Europe, and when you look at the east coast of the United States, you see darker colors, which indicate a greater incidence and introduction of more invasive organisms. This is often related, as we know now, to shipping routes, which is that map that's demonstrated with all those lines, which shows the connectivity that we now know so well that connects us all. And when we look at the map of Europe, again, this demonstrates the introduction of invasive species throughout Europe. The size of the circles denotes the number of species, and the colors denote the various mechanisms. And you can see that throughout most of Europe in marine environments, most invasive species have been introduced either via shipping, which is the green color, or via aquaculture, which is the orange color. There's a slight variation on this in the Mediterranean, where we also see the impact of the Suez Canal. You can see that in the yellow in the larger circle at the end. And this is particularly related to movement of organisms through the canal, but also again through those vast shipping routes. When we look at invasive species and why they are able to move around via these different modes of transportation, either shipping or aquaculture in particular, we see that often these organisms are successful in their new habitats, mainly because they have a number of general characteristics. Often they have few natural predators, competitors, parasites or diseases in their new habitat. They often have high reproductive rates that can give them more success in relation to native organisms. They're often long-lived and they're generalists and they're pioneer species. They can adapt to the new environments very quickly that they find themselves in. And when we look at the potential role of these invasive species in the introduction of disease, there's a number of different scenarios that you can look at. Sometimes these organisms that are being moved around and transported are often infected with parasites. We see that quite a lot in aquaculture. So organisms can be moved around and they're already infected. They may have few or no parasites, which can often be why they are so successful. They may not bring their parasites with them. When they establish themselves in a new area, they're often not susceptible to the parasites or pathogens that are present in their new range, giving them a competitive element or and factor over the native species. Or sometimes they can even be parasites or potential pathogens themselves. And we've been particularly interested in knowing that can some of these act as incidental carriers or reservoirs of pathogenic organisms or parasites, and so be a potential route for carrying new diseases through the marine environment. When we look at the marine environment and when we look at the risks that are associated with invasive species, we often look at them under three main tenets or factors. There are those environmental risks associated with invasive species, there are the economic risks, and there are the wildlife and public health risks. But the three common denominators between these three elements are the risks associated with parasites and with diseases. And we see that in the different tenets. So we find that the potential impact of parasites and diseases can be one of the key risks associated with the spread of invasive species. And how do we actually determine what level of risk that might be? And a seminal paper by Williamson and Fisher in 1996 introduced the concept of the rule of tens. And when we look at this schematic here, it basically demonstrates that if you have potentially maybe 100 introduced species, of that 100, maybe 10 will become established. And of that 10, one invasive species may become established and invasive out of that initial 100. We see similar levels of potential risk associated with the parasites and diseases that might be associated with these invasive species. So it gives us an idea that a lot of organisms have to move around, and the risk associated with that will be a factor of the number of that starting number of organisms. Now, a lot of our work in our team here at University College Cork has really focused on the impacts of pathogens and diseases on aquaculture species. A lot of our work is on shellfish diseases. It's on the pathogens and the diseases that have often mainly been introduced into Ireland with those organisms. And more recently, we've been looking at how these different pathogens can actually spread out into the environment and how they can be taken up by other marine organisms, having wider ecological impacts, but also being picked up and becoming being carried around by organisms that themselves may be transported around or may be invasive species. And for instance, we've looked at things like Carcinus, the crab, which is invasive in other habitats and in other countries, and in things like the tunicates, which are, you know, a major problem as an invasive species in terms of their impacts on aquaculture. So just to give you an idea of how we are thinking about this question, one particular pathogen and disease we've been working on a long time is Bonemia austria. It's a protystin parasite that infects the native flat oyster, Austria edulis. And the thing about Bonemia is that it infects the blood cells of this oyster, but transmission can occur directly from oyster to oyster. But we also know now from work that we've done and other teams have done over a range of and a period of time is that these parasites can survive in seawater. And this schematic just shows that when the environmental conditions are optimal, particularly when there are optimal temperature and salinity factors, that parasite survival in seawater can be a week or more. So these parasites can be transmitted in the seawater if those conditions are right. We've done quite a bit of work showing then that this parasite and some of the other main pathogens can be transmitted within environments like the internal shell cavity of mollusks and can survive there for quite a period of time, even in organisms that are not natural hosts for those pathogens. And we've also been able to show in a range of studies that Bonemia, for instance, can be found in a range of other organisms, such as zooplankton, things like brittle stars, and in a whole range of tunicates and sea squirts that are in themselves invasive species. And we know that these range of organisms, some of them actually can act as carriers or transmitters of these parasites. So potentially we find that the spread of these parasites into other organisms can result in the movement of those parasites via these incidental carriers. We've recently been working on an EU-funded project, a European Regional Development Fund project between ourselves and a range of partners in Wales. This is led by Bangor University in Wales, a project called Bluefish. It's looking at climate change impacts in the Irish Sea area. And we've been working on ecosystem health, and we have been responsible for looking at the potential impacts of invasive species on the Irish Sea area over the next 50 to 100 years. And these are two of our PhD students, Katie and Sarah, who've been working on this project. Katie on the left has been responsible for the work on invasive species. She's been looking at the importance of marine bivalves in invasive host parasite introductions, the role of invasive tunicates as reservoirs or carriers of some pathogens, and has also been looking at assessing the potential for invasive species introductions and secondary spread using vessel movement in maritime ports. And in some of her initial work, she's shown that we have over 51 invasive species in Ireland, a range of different species, many of them which can have significant ecological impacts, things like Crepidula and some of the tunicates. And this map just shows the amount of ballast discharge that occurred in Ireland. This is quite an old map, and there's even more significant levels still. And these species that have been introduced from sources worldwide via our shipping channels. In some of her work, which was a review of what we know to date around the importance of marine bivalves in invasive host parasite introductions, she's been looking at how invasive species and invasive hosts could transmit parasites and have an impact on native species. And this schematic just really shows that when invasive species are brought in and bring parasites with them, they can have a whole range of different impacts on the native hosts and on the native habitat, either via bringing parasites in themselves, or transmitting parasites to native hosts, or even picking up parasites that are present in the native range and actually amplifying the impact of parasites in that region. And our review also shows that climate is going to have a more and more significant impact on the impact of invasives in regions due to impacts on the pathogens themselves, on the host, and changes in the environment. So more and more vigilance is going to be needed in terms of the increased risk associated with invasives over the coming years. She also looked at a particular model species, and Katie looked at invasive tunicates, and this is Styla clava, which has been introduced to Ireland. It can be a fouling organism on aquaculture and here on oyster bags. And she's been looking at that as a reservoir of some of our mollusk pathogens. So Katie carried this work out in a few different regions in Ireland, but also in the lab as well. So she carried out a range of field studies where she looked at some of our oyster species. She looked at some of the, a range of the parasites that we find in these native oysters. And then she screened a whole range of tunicate species that have been introduced into Ireland. And what she was able to find is that these tunicates had picked up and were acting as carriers or reservoirs of a range of protistan, of viruses and bacteria that have been introduced to Ireland via our aquaculture species. And because of the risk associated with the transport and the movement of these tunicates, these could be a vessel for further transport and movement of these key pathogenic species. As I said, she was also looking at the impacts of the movement and spread of invasive species via shipping channels in Ireland. And she looked at over 9,000 shipping movements into Ireland over 2 years. This shows our shipping routes out of Ireland. And what she found when she looked at these 9,000 shipping movements is that they come from a whole range of different shipping routes throughout Europe. And we have a year-round movement of ships in and out of Ireland, so there is no significant seasonal component to this. She was also able to look at the types of ships coming in and out, the times in ports, and the travel time, to really try and determine if these shipping routes were a good opportunity for invasive species to be transported into Ireland, and if they would be viable and be alive when they arrived in Ireland. And we have a whole range of invasive species that have been identified as potential risks in terms of introductions into Ireland. She picked out 5 key species that are considered of high risk of moving into Ireland. These are mainly crab species, razor clams, and crepidula. And she also did some background information and found out what parasites do these particular organisms normally have. And these different potential invasive species are infected by a whole range of different types of parasites that we don't necessarily have in Ireland at the moment, but have wider impacts on a range of species when they're introduced into an area. So what she did was, when we looked at all the key ports from in a range of different countries, when we look at these dark blue areas, these are ports where these invasive species are currently present and where there is many movements of ships from those ports into Ireland. So the risks and the opportunities for these invasive species to be transported, potentially with their parasites, into Ireland are significantly high. And when ships come to Ireland, there's often quite a lot of secondary movement of goods and materials and ships themselves from the port of entry to a range of ports around Ireland. And we identified that this is resulting in the potential for secondary spread of invasive species from these original ports is quite high in Ireland. So the results overall of these studies found that 4 ports in Ireland demonstrate high connectivity. There are multiple routes of entry for potential invasive species. Many of the connections are to ports in which invasive species are confirmed to be present. Invasive species that are themselves infected with a range of pathogens, and that these focal species produce multiple broods per Ireland, which gives a wide temporal window for larval stages to be taken up by vessels and transported, along with adult stages. The short journey time between ports also facilitates the journey of potentially pathogenic microorganisms that might be associated with these invasive species. The high connectivity around Ireland, and this is similar to a range of other countries, means that secondary spread is very likely, and that ships arriving from a couple of particular countries are potentially high risk due to the high shipping volumes and the confirmed presence of invasives in the regions. So to finish up, this has been a very brief overview, but mitigation strategies for invasive species and their potential role in disease transmission are multifaceted. And I suppose I would be emphasizing today that all stakeholders, all of us that engage with the marine environment, have to be vigilant, not just of the potential impact of invasive species themselves, but their potential role as reservoirs or hosts or carriers of potential pathogenic organisms that can have much wider ecological, economic impacts if they're introduced into new areas. So thank you very much for your attention. And I'd like to particularly acknowledge Dr. Sharon Lynch, my collaborator, and Katie Costello, PhD student. Thank you very much.